Everybody here? Are we ready? No? Welcome, one and all, to the Potomac Institute. This is a room full of friends who have all been here before, I think. Welcome to the second in our series of bold idea talks here at the Potomac Institute. We have embarked on a mission of finding the most enlightened, thoughtful people in our society to talk about big ideas that can address big problems that our world and society faces. This is hard to do. Lots of the best people actually aren't trained, skilled, or have a great background in thinking out of the box and thinking about the big ideas. One of the most exciting parts of my job at the Potomac Institute is being able to be associated with, from time to time, people who really do that. People who have groomed themselves over a career to not just worry about things in their discipline, worry about things that were in front of them from day to day, but to take the time to worry about our world, our society, and how science and technology might play a role in helping our society and our world be better. So, Bold Ideas seminars are about bringing those people to this room, documenting a presentation, and publishing it and helping to spread bold ideas around this town and, in fact, when we can, around the world. Today we have one of the most broad thinkers I think this town has ever seen. And before I introduce her, though, I want to announce that we're going to have another seminar on September the 20th. Dr. Victoria Keener from is going to talk about climate change impacts in the Pacific. She's from an institute in the Pacific in Hawaii who have been studying these issues. They're in town to brief Congress on climate change in the Pacific and the impacts of climate change on society and, in fact, national security. So she'll be here in a couple weeks, and we welcome you all back for that one as well. But today, as I said, we have someone who's been my privilege to know for quite some time, and to tell you that this person has probably inspired me as much or more than anybody I've ever met in Washington, D.C. as a scientist who addresses the issues of national security from a broad perspective. Very few scientists that I know have the wherewithal, the thought process to understand that science drives national security, drives our societal security as much or more than anything in the world. So I didn't actually intend to introduce her. I'm not. I'm going to ask Professor Dennis McBride, our President Emeritus of the Potomac Institute, to introduce Rita. But I did want to highlight for you that she has been an inspiration to the Institute and I think to this town. And I think you're in for a real treat today. Dennis?
Thank you, Mike. Rita. It's such an honor and a pleasure to introduce our featured speaker here. You know, they say some people don't need an introduction, and Rita Colwell is one of those who does not need an introduction. The problem is I'm Irish and I have a captive audience, and therefore you're going to hear an introduction. I think everyone in the room has known or known of Dr. Colwell for many years. Probably outstanding most in our minds is her leadership of the National Science Foundation, and what a series of years. I don't know if NSF has ever grown and done as well as it did under her leadership. There's some things about Dr. Colwell that you might know and you might not know. She is now, of course, a distinguished university professor at the University of Maryland, and she is also a member of the Bloomberg School of Public Health at Johns Hopkins University. But her depth and breadth go back quite a long ways. Dr. Colwell has produced, count them, 56 PhDs, 56 under her direct tutelage. I don't know of any two professors. I'm Associate Vice President for Research at George Mason University, and I know a lot of professors in a lot of places. I don't know two who've produced that many PhDs. But it goes beyond that. 3 of her PhDs are National Academies, and the odds are that would never happen from a set of 56 tutored PhDs in your lifetime. And a couple of other interesting personal stories about Dr. Colwell. is, and I just love this one. We were having a conversation one day when she lived next door at the National Science Foundation, and she was had just ended or was ending her tour there, and she was going through some old materials, and she found something that she had jotted down when she accepted the job to go to National Science Foundation, and I guess she put it on her refrigerator, as we all do, a to-do list. And I think it surprised her in this box. She pulled it out and she went through the list, and darn if she had not gone checklist complete. She had been through everything on her list and had forgotten about the list, but managed to accomplish it anyway. Hell, I can't get halfway through a list that I write down, and I keep the list with me. This woman had accomplished all that and more at the National Science Foundation. So as a world-famous microbiologist, and I cannot wait to hear what she delivers to us today, I'll close by saying, despite everything you hear about Dr. Colwell, if you met her at the bookstore and you had a pleasant conversation with her, you'd say, you know, I just met one of the nicest people that I would ever meet, and not know about her fame and her glory and her contributions to our national situation and our international knowledge of her specialty area. So Dr. Colwell, without further ado, I turn the microphone over to you.
Thank you very much, Dennis and Mike, for the very kind introduction. Is the mic getting you at the back of the room? Good. Thank you very much. It's a pleasure to speak here at the Potomac Institute. I've been associated with it, gosh, for more than 10 years, and it's been really a pleasure and very exciting to learn about the new studies that are being contemplated, and of course I'm familiar with the ones that have been done. What I'd like to talk to you today is about the potential role of infectious disease influencing human health in an era of climate change. So what I will start out doing is, first of all, describing a little bit of background about the one disease I know a lot about, and that's cholera, because it's a waterborne disease. And as I give you just some of the background facts, including some of the genomics, I will then launch into how we can predict cholera outbreaks based on climate events, including some results that we have for Haiti and the current situation in that country. Now, the general background of what I have to say is nicely encapsulated by Hippocrates, who wrote in the 4th century BC that whomever wishes to pursue the science of medicine must first investigate the seasons of the year and what occurs in them. In other words, the seasonality of diseases like cholera and influenza have a lot to do with the bacterium, or in the case of the virus, and in the case of the virus, its proclivity to lower temperatures and becoming more infectious. In other words, the relationship to the seasons of the year in which these diseases occur. Very clearly, the effect of water and its safety is very amply demonstrated by the effect on children. Those under the ages of 5, in the case of those that on the left, Cambodia, Ethiopia, Zaire, don't have access to safe water. That is, less than 20% of the population has access to safe water. And the result is that 200 of every 1,000 children under the age of 5 will die from a diarrheal disease. So these are very dramatic correlations. And I will discuss cholera because it is a global disease. It's an acute water-related disease. We talk about pandemics, but that's because it's cyclical, and the data that we have begin really in the recorded outbreaks about 1823, and there are periods when cholera sweeps through the country, the U.S., Europe. We unfairly consider India and Bangladesh as the home of cholera, but really the U.S., New York, Washington, Boston were the places of epidemics in 1848, 1880, 1900, until the introduction of safe water. In fact, at that time, diseases like malaria and cholera were presumed to be caused by miasma, bad air, and Washington was considered a miasmic swamp. Some of us think it still is, but different reasons. More than 50 countries Every year, about 7 million people are affected by this disease. And as I say, we've considered the Bengal Delta as the home, but rather unfairly. It's just that there is where it is no longer controlled. It exists, the bacterium. We showed the work that my students and I were able to do over the last 25, 30 years, showed clearly the bacterium is part of the aquatic environment and that it cannot be eradicated. It does too many things for us in terms of ecosystem services, fixing nitrogen in some cases, breaking down hydrocarbons. In fact, one of the Deep Horizon findings is an increase in vibrios, Vibrio cholerae being one of them. So it breaks down hydrocarbons and it plays a role in the carbon cycle in nature. So it's unlikely that we'll be able to control it. The bacterium, for those of you who are not microbiologists, it's a bacterium that has a single flagellum that allows it to be highly motile, and it attaches to the villus of the intestine and produces a toxin that interferes with the sodium-potassium transport, and the result is a loss of up to 17 or 18 liters of fluid, dehydration, and systemic shock and death if not treated. We discovered that it goes into a dormant stage, which is kind of interesting because it means it can, during the winter months, low temperature, low nutrients, it can't be cultured, but it's still alive. It doesn't produce a spore coat, but it persists in the environment. And the fact that it can be detected using molecular techniques now, we used monoclonal antibody 25, 30 years ago when we discovered this phenomenon, but now with gene probes, particularly probes that are fluorescently labeled, you can detect quite accurately the presence of Vibrio cholerae in the environment. And its relationship with plankton is, of course, critical. And that's one of the reasons why it cannot be eradicated. It's part of the commensal flora of the copepod, and that represents the most dominant species in the world oceans. So this bacterium is distributed widely throughout the world. We could detect in Bangladesh, by just doing that fluorescent stain on a copepod, that up to several hundred bacteria, cholera bacteria per copepod, can be detected. And then in the spring and the fall, when the copepods respond to first the phytoplankton bloom from sunlight and increased temperatures, and then the copepod graze on the phytoplankton, they become abundant, and then thereafter the cholera epidemics occur. It's like clockwork, unfortunately. Spring and fall, epidemics, small in the spring and a major one in the fall, in Bangladesh, in India, and other developed countries. Lots of sources of the bacterium, but predominantly the one in the red in the center, the copepod, is dominant. So this bacterium, these vibrios, play a major role in the environment, providing services for us as humans. Summarizing this introductory part of my talk, these are the sources in the environment. Certainly, the pollution engendered by poverty and lack of sanitation, lack of safe water. There's person-to-person transmission, no doubt. That amplifies the disease imminently, just as we've noticed in Haiti. But clearly, the source is the environment, and that has to be understood. Now, we've developed a model based on the ground truth data of about 30 years of working in Bangladesh, measuring temperature, salinity, nutrients, all of the parameters we could possibly gather, and then building a model showing that with sunlight, increased temperature in the spring, the simple model said, yes, there's a phytoplankton bloom, then the zooplankton graze on the phytoplankton, along with cladocerans and some of the invertebrate larval stages of animals. And then subsequently, there's a release into the water column with the infectious dose elevated so that anyone drinking the water, non-filtered, without any treatment, would then succumb to the disease, particularly children. And with that simple model, we were able, 15 years ago, to show a direct correlation with the red being the predicted based on the model, and the white, the columns, the actual cases in Matlab, Bangladesh, where we have done most of our work. Now, now we've been looking at the macro scale processes. We know from previous work that rainfall, flooding, how high or how low the river will be during times of the year, sea surface temperature, salinity, fecal contamination, these are all factors that play a role. But how can we drive this in a model on a global scale? Because we know that the outbreaks, more heavily darker the color, the more serious the outbreaks, Africa, and then the 1991-92 massive epidemic with thousands dying from cholera outbreaks that occurred during that El Nino relationship, which we were able to show and publish. But we've focused predominantly in at least in the literature, people have studied India, Bangladesh, the Bay of Bengal, the relationship of the Himalayas, the river system, and the delta itself in the incidence of cholera. And we've done most of our work in MATLAB, which is a kind of a province, if you will, outside of Dhaka, sort of southeast a bit of Dhaka, the capital city, and worked in the hot spots in red around the ponds which are excavated, and on the earth that's excavated are where the houses are built, and then the ponds used as a source of drinking water, washing, and unfortunately one corner will be the latrine. Bacteria don't behave themselves and stay in the corner, so you have this transmission cycle going on. But we've been able to show the relationship to sea surface temperature. This has been quite critical because as the temperature rises, and you see here NASA downloaded data for multi-year periods, and the sea surface height plays a role too because it's the incursion with the tide and tidal flows into the river systems that are significant, bringing the plankton in and causing the coastal area epidemics. But it's the plankton that, of course, drive the outbreaks, and this is related to these factors. Now, taking that into account, it occurred to us that we could use satellites to measure and predict cholera epidemics. And so, in fact, one of the studies we did very early on, about 15 years ago now, almost 20 years ago, we simply took data downloaded again from the NASA headquarters that the NASA labs that we worked with for the Bay of Bengal. And quite interestingly, we found that simply taking sea surface temperature and the numbers of cases of cholera going into the MATLAB hospital, the correlation was quite dramatic when you consider we were simply using a model of our ground truth data, satellite imaging, and then just using the satellite sea surface temperature and the numbers of cases in MATLAB Bangladesh, and getting a rather dramatic correlation, which we published in PNAS about 15 years ago. But now we've been making a much more sophisticated approach in understanding the epidemics, and we've been studying in Kolkata, as well as MATLAB Bangladesh, and we've been able to relate very significant relationship between cholera, chlorophyll, and rainfall. These have proven to be quite significant. In fact, in Calcutta, with a milligram per cubic meter increase in chlorophyll in the sea surface, we can anticipate about a third increase in the number of cholera cases in Bangladesh. And with a millimeter per day increase in rainfall, we can predict about a 7% increase in the number of cases. Correlation on a macro scale of these climate parameters with the outbreak of cholera. And similarly, same results in Bangladesh with a milligram per cubic meter increase in chlorophyll with about a third increase in the number of cholera cases. So now we are looking the next step, because we know that phytoplankton plays a role because of the relationship of the zooplankton to feeding on the phytoplankton and the cholera bacteria. We can estimate over very large areas using the satellite-derived chlorophyll measurements And we know from all of our laboratory work and our field work the fact that the bacterium requires salt for maintaining its cell structure. In fact, one of the early studies by Robert Koch in 1890 was a test he used called the string test. It was taking collected cells, centrifuged, putting them in a test tube, adding distilled water, and then adding a sterile loop, and you get all this gunky stuff. Well, that's because we now know the cells were lysing, and that gunky stuff was DNA. So the relationship then to salinity has been quite dramatically demonstrated, as well as the chlorophyll estimates we've been able to do. And the chlorophyll estimates are about 60% accurate, and we're now seeking to get even greater accuracy in our prediction. The recent work we've done, we've been able to separate the spring epidemic, because it's much more related to drought and reduced water flow, and then the fall epidemic is quite different because it's related to the monsoon, the flooding, and the effects of the flooding. And we've been able to show that we can determine how large the spring epidemic will be based on how intense the fall epidemic, hence the relationship to the severe weather events, the heavy flooding that occurs in Bangladesh. And so we've been able to predict, using a model, the spring and the intensity and the timing of the spring and fall epidemics. So we're now trying to utilize the large-scale hydroclimatological processes to be able to do this prediction. And we're now currently developing not only the hydroclimatic forcing, but we're also taking into account the immunological resistance of the population and the transmission of the disease person to person, those kinds of data that come in from understanding the infectious disease itself and the immunological response of the individuals. Now let me just for a minute step aside and talk about the genetics, because this is also very important in this multi-dimensional, from the molecular structure of the bacterium to its environment and to the large weather processes. We, in the year 2000, which is now 13 years ago, time does fly, we sequenced for the first time the cholera bacterium and confirmed what had been hypothesized, that it had 2 chromosomes. Until then, it was assumed all bacteria had a single chromosome. And we found that there's a large chromosome and a smaller chromosome. And we were able to show clearly the smaller chromosome was a chromosome and not just a plasmid or ancillary DNA, because we found that the genes on the small chromosome had a whole lot of housekeeping metabolic functions that, if deleted, the bacterium wouldn't function. Now, since then, we've sequenced nearly 200 isolates from all over the world. And historical strains isolated in 2010, strains isolated very recently, strains from India, Bangladesh, and most recently from Haiti. What we found was that there is no strain-to-strain identity. In other words, what we've done here is kind of take the chromosome of the original, and that's in the pink and orange at the top, and stretch it out. And then we've done the same with the large chromosome and the small chromosome and matched them, base pair by base pair. And where you see blue means that that particular base pair or combination of base pairs is missing. So you can see that there's a great deal of variation that occurs just within a single species, and that this can be used to allow us to track origin of the epidemic, not only country to country, but as we are now showing and just getting ready to publish, that the epidemic arises locally. In other words, the genome of a bacterium, cholera bacterium in pond X, where people are taking their drinking water, and pond Y, the genomes are different. And they are from the environment, and that this then is a locally introduced disease. We've been able to track the evolution of the bacterium, and this has proven to be very important because one of the things we discovered was that the genes coding for the serotype are laterally transferred. And that means for 100 years we've been using serology to identify the epidemic strain. But in fact, what we've been doing is tracking the genes that code for that serotype rather than the whole bacterium, which may explain why we've had difficulty getting a vaccine. Because the vaccine, if we get one strain that has the serotype genes and we get a vaccine to it, but those genes coding for serotype are transferred to another strain, then that vaccine won't be as effective against that strain. So we've since found that almost 90% of the genes in this very versatile bacterium can be laterally transferred. It's a very interesting bacterium. It is a human pathogen, probably because we are an inadvertent host. In other words, it behaves, if you will, just for to give an example, as if we are one great big copepod, and we're trying to do whatever we do, probably, my hypothesis, osmoregulation. But in osmoregulating our gut, we end up with severe diarrhea, vomiting, dehydration, and if not treated with rehydration, death. But in the copepod, it allows it to migrate, for example, in the Chesapeake Bay, from oceanic salinity all the way up to Baltimore Harbor. And that's a difference of 30 parts per thousand to a half a part per thousand salinity. Now, we now know that with this gene transfer bit, we need to look at cholera, a bacterium, the way we look at HIV virus and influenza virus, namely that there's genome drift and genome shift, variations that occur which make it difficult to have 100% vaccine effectiveness. Now, the other surprise that we got was, being NSF director, I got to do some wonderful things. It's a great agency, terrific people, and I'm not saying that just because some of you are in the audience. I really, really enjoyed my time at NSF. I got to do a dive on Alvin. And of course, someone asked me, weren't you frightened? I said, no way. Woods Hole was not going to have a headline in the Washington Post or the New York Times, NSF director lost at sea in Alvin, so I knew I was safe. But one of the things I observed was, and I should have known, were these things swimming around the hydrothermal vent. And I asked my fellow scientists, there are 3 of you in Alvin, the pilot and 2 scientists, so you want the pilot to be in very good health, that indeed there are deep sea copepods. So I asked for a sample, got a sample. We isolated bacteria from the deep sea copepods. We found that from this location in the East Pacific Rise, 2,500 meters down, we found that these vibrios carry a whole lot of the same genes as Vibrio cholerae, Vibrio parahemolyticus, and Vibrio alginolyticus, all human pathogens. What's a deep sea Vibrio doing carrying pathogenicity traits? Well, the fact of the matter is that, just going back to the Venn diagrams here, the genes that we ascribe pathogenicity traits are really fitness traits for the bacterium in the environment. And we need to decode what it is they do in the environment, because that can illuminate what they are doing as a function in the human body. Now, another example of, you may think I'm sort of overdoing this, but it's important to understand this bacterium is ubiquitous in the estuarine, marine, and even in the freshwater environments where there are copepods, cladocerans, and its normal hosts. In Iceland, they have never reported cholera. It's never been reported, but they have a superb health registry. And in fact, they can track back to the founding of Iceland diseases that occurred at that time. And cholera is reportable and they have other cases of pandemics like plague and influenza. But in these areas that are geothermally influenced, particularly where the water temperature can rise above 30 degrees, but that at low tide, and then when it mixes with seawater, the temperature goes down, and so there's this fluctuation. Graduate student, who is now Dr. Brad Haley at the USDA, did a Fulbright in Iceland sampling the geothermal sites. And he found, as you can see, all these pluses, that Vibrio cholerae, the causative agent of cholera, could be isolated from Iceland. Now, what's a self-respecting cholera bacterium doing in the waters of Iceland? It's because it's a naturally occurring bacterium. It's an estuarine bacterium. It's globally distributed. And similarly, we were able to find that many of the genes that we call pathogenicity genes are present in this bacterium. Some of the toxin genes were not present, but many of the other pathogenicity genes are similarly present in those cholera bacteria in Iceland. Now, why is there no cholera in Iceland? First of all, drinking water, those of you who have traveled to Iceland know that it's essentially water that's distilled because it's geothermally heated. It's, by the time it gets to the household, it's essentially boiled water. They don't typically eat shellfish. They use it for bait. And they don't have much interaction. They don't swim in the waters. They have swimming pools that are geothermally heated. But I think it's pretty cold to swim in the water beaches of Iceland. And why do they have so many of these virulence factors? Probably because they play a role in the ecology of the bacterium. For example, sialic acid genes have been associated with disease in humans. Well, it turns out mussels, which are abundant in Iceland, have a high concentration of sialic acid, probably why they have the enzymes and the function for sialic utilization. Now, let me turn to taking all of these data gathered over 35, 40 years with many terrific students. In fact, I like to say that all of my students are gainfully employed, none's in jail. That's a good record. These students have been terrific, and what we've done is take all of this work, the genomics, the prediction, the satellite data, the weather, extreme weathers, to see if we could actually link together what's happening in Haiti with extreme events. The outbreak occurred near Port-au-Prince, but it very, very rapidly, from that center, supposedly the original contamination caused by the UN peacekeepers, according to the newspapers, but simultaneously, these outbreaks were occurring throughout the entire country. And working with Anta Jutla and Ali Akanda, two hydrographical engineers from Tufts University, working with Shafiq Islam, a professor, an engineer there. These two guys are engineers. They're hydrological engineers. And for the river to flow down to those areas where the outbreaks to occur, the river doesn't flow that fast. So this simultaneous outbreak all around the country hydrologically cannot be explained. And the outbreaks were occurring in each one of the arrondissements of Haiti. We were able, within the first few weeks, to get, by collaboration with a physician, stool samples cultured. We shipped in because they don't have laboratories. They claim cholera has never been reported in Haiti, but they've never tested for it. How can you claim if you've never done the testing? They've had severe diarrheal disease, and the hospital records show that. So we were able to get from 80 patients, we were able to, 81 patients, to get samples. We shipped in all the, because we do so much field work, we shipped in everything needed. The filtration units, the petri dishes, the sterile test tubes, the swabs, everything. Shipped it in to Dr. Prosper a colleague who was able then to sample the patients and isolate cultures, which the samples were sent back to us, and from every petri dish we isolated hundreds of colonies. We did far more work than you would normally do in order to be certain of what was happening. To make a long story short, and 2 years' work in a minute, we found that, in fact, genomically, there's a separation of the Haitian strains, and I won't go into the details, but using OpGen approaches and very high sophisticated approaches, there are variations in the base pairs of the genes coding for the toxin and genes coding for some of the other factors. And principal component analysis shows that there's the Haitian cluster, and these are strains from India, Bangladesh, Zambia. In fact, the analysis clearly shows that the Haitian strains formed a cloud distant from the other genomes. And the strain that was used in the initial publication, the strain that we had isolated and sequenced, no, we didn't isolate it, we sequenced it in year 2000 that I showed you the results. That was the strain that was matched by the Centers of Disease Control using a very elementary approach. It's not elementary, I should say, a less precise approach than sequencing, to say that it was of Asian origin, and therefore the conclusion was rapidly drawn that it was the Nepal troops that brought cholera to Haiti. But it was more distant than a strain from Zambia, Thailand, from the Haitian strains. So the genomic analysis that we were able to do provided two distinct Vibrio populations, because from some of the patients, half of them, we did not isolate, could not isolate the cholera Vibrio. We isolated a non-O1, that serotype. Remember I told you about the genes coding moved around? Well, it was only strains that didn't carry the serotype genes. And the serotype genes, of course, are the ones that are used to say that this is the cholera that was imported. So our conclusion was perhaps the Asian strain was imported, but it's much more complicated. The strains that are already there participated in the outbreak, and some, about 7% of that 81 patients, didn't carry Vibrios at all, but Aeromonas and other enteropathogenic strains. So our conclusion is it's far more complex, and rather than jumping to a conclusion, let us try to understand the ecology and the epidemiology. Now, let me just go on to another aspect, and that is we were able to get the data from India from about 1823 to 1948. And with a collaboration of a wonderful fellow scientist, Dr. Elizabeth Whitcomb from New Zealand, who for many years ran a hospital in London, who was had done a PhD in India on the hydrography and the relationship to malaria and diarrheal disease back 25, 30 years ago. And then had gone on to do a medical degree, so she's an MD, PhD. The PhD was from Oxford, and the medical degree is from University of Auckland in New Zealand. She had discovered these maps, about a third the size of this table, that reside, and that you can lift up, there's just map after map, with meticulously plotted every cholera death and every malaria death. They called it fever and diarrhea, because we're talking about 1823 to 1948. And so we computerized, put everything on, all of those data on the computer. And then we also were able to obtain the meteorological data, rainfall, temperatures, air temperature, and so forth. And we were able to show, when you plotted the data for the 9 locations in India, there was a very clear correlation that when the temperature was elevated, there was a relationship to the outbreaks of cholera. Furthermore, we were able to show the relationship using a normalization approach between, for Delhi, Lahore, and so forth, all of these cities, the relationship between the cholera outbreak and the season of the year when the epidemics occurred. And we were able to show with an odds ratio analysis that the odds of cholera when the temperature was above average, you can see in red, and when the temperature is below, you can see in blue. So we were able to tease out this relationship of air temperature 2 months before the massive rainfall. And we were able to show similarly in Haiti that what we have concluded to have occurred in this country, first an earthquake, massive earthquake in the spring of 2010 in January, a country that already had very bad sanitary conditions, very little access to safe water, and then the hottest summer on record, and then the heaviest rainfall in 60 years. When I say on record, 60 years, and again, the rainfall. And so the correlations, let me just move on to, led us to the following model, that when the rainfall is above average in a poor or damaged water and sanitation situation, very high risk of a cholera outbreak. And I should point out, above average temperature for 2 months prior to the outbreak. This was a model we developed from the India data, the massive data analysis for India. And the air temperature in Haiti, 2010, and the rainfall, 2010. So in essence, what we had were the 3 horses of the apocalypse, or maybe the perfect storm, that then resulted in the massive cholera epidemic in Haiti. The other aspect that I should mention is that the geology of Haiti is such that it's alkaline structure. And with the grinding of the earthquake, the river's pH went up to 8.6. We've measured the pH of the Atabaniti River, and it's 8.6 pretty much down to the mouth. And how you isolate Vibrio cholera when it's in the environment, you take a sample, you put it into a very high alkaline, 8.6 to 8.9, elevated temperature, incubation for 24 hours, and you'll get explosive growth of the bacterium. So using the laboratory information, the genomics data, and then the weather data, we're able to put all this together to develop an hypothesis of that epidemic. Now, I've been talking about a part of the world, the Bay of Bengal, where you can see the population increasing from 1,700 to 2,000. And this is some wonderful data that Tom Paris and Matt Luck and Rick Saccone provided for me through a Medea study we've been doing. And this population density, as you see, we're now getting to 1960, 1970, 1980, 1990, 2000, and it would be even more red coloration if we carried it to 2013. This is a heavily populated area with a watershed that we know from the Gangetic in the Brahmaputra Delta and the relationship to the Himalaya watershed, with intense water reuse. But it's also an area that the climate change predictions are really very severe. The red is with a 1 meter rise, but I can tell you that those are areas that are now almost constantly underwater. As you get to the gray area, that's a 7 to 8 meter rise. So this is a part of the world where a country with, I think, 250 million people in the area of maybe South Dakota or North Dakota, these are going to be climate change refugees that are going to be migrating to other parts of Europe, the U.S. And the disease potential will be severe with that kind of flooding, with increased temperature, and with populations that have to deal with flooding conditions now annually, but very likely to become much more severe in the future. So I'm going to close by using the words of John Muir, who said, when one tugs at a single thing in nature, he, and I would add, and she, finds it hitched to the rest of the universe. And finally, I would like to thank collaborators from the International Center for Diarrheal Diseases Research in Bangladesh, the many students, postdocs, colleagues from NIH, from NASA, from ESRI, and even from the intelligence agencies, all who have worked with us. Thank you very much. Glad to take any questions. Yes.
It's a fantastic talk. Thank you so much for that. It's incredible work. I was also wondering, in terms of this relationship epidemiology to the warming trends and so on, if you look back, let's say, in the mid-10th or 13th century, in the medieval warming period, were there correlations, do you know, with other outbreaks and diseases and so on? In other words, was there another period of time, setting aside our current challenge with climate change and so on, where this also correlates?
The question is, how do you correlate from earlier seasons, earlier times like the 1400s or 1300s with disease outbreaks? We're actually doing this with plague because we've been collaborating with a team at the University of Oslo in Norway and doing work in Tbilisi, Georgia, where they have plague every year, not a whole lot, but they have outbreaks of it because it's naturally occurring. And looking at and in this case, it's, I guess, gerbils and rodents who are the carriers. And it's a relationship that can be plotted, outbreaks, with climate. And it also, we're beginning to have data showing that the Yersinia pestis that causes plague undergoes a kind of dormancy as well. And that, which makes sense because in the case of cholera in the copepod, the copepod undergoes it burrows in the sediment in the winter and it goes into a dormant state itself. So it makes sense that the bacterium associated with it would do the same. And similarly with these rodents, they burrow during the winter months and then the outbreaks are very seasonal. And we're relating the temperature and climate of plague cases that occur in China and in Georgia, in Azerbaijan, with the climate factors. And so we're doing that now with some interesting correlations of earlier outbreaks. In fact, even, sounds gross, but digging up mounds of burial victims and being able to isolate and doing the genomics on the original isolates to get a sense of what kind of variations have occurred genomically as well. So it's a fascinating area of study right now. Yes. Yes.
Rita, thank you very much. An observation and a question. The observation is that using some of the same genomic techniques that you are using, tuberculosis has been similarly tracked.
Yes.
found to have originated in Africa and spread with the human species, and then has recently gone back to Africa, and also has evolved into a form now that is much less likely to go into a resistant stage and stay there, but rather remain highly viable in populations. My question, you stated that a dose of 10 to the 6, greater than 10 to the 6 cells per milliliter caused the disease. Has there been work on looking at ways to regulate our salts in our water in the human body in order that 10 to the 6 becomes a small dose?
Well, the better way to regulate is one that we suggested based on all of this work, and that's a very simple filtration technique which we introduced in India. The question was how can you reduce, if you change the body composition or salt fluid composition, if you'd be less susceptible. The fact is you can eliminate the bacterium, which is what we've done. I mean, none of us in this room, I think, I've had the cholera vaccine, but none of us takes it regularly because we don't have cholera, because the water that we drink you use alum to coagulate it, it's filtered, it's chlorinated, it's safely transmitted and distributed, so we just don't have to deal with it. In the case of the cholera bacterium, it's another good point that it was made, that I pointed out that about 1 million per milliliter, 1 million per teaspoon of water, whereas there are some bacteria like Salmonella and Shigella, all you need is 1 or 2 cells. So the virulence can vary. And with cholera, we found in doing this filtration study, where we were funded to go in to do a 3-year study to educate women to just use simple cloth, sari cloth, to filter the water. We reduced the cholera by 50%, but we found also that that the flamboyant case of cholera, if you will, dropped, and the mild cases, because you don't get all the bacteria out, but when you have just a small dose, you might have an episode of diarrhea, and that's it. So you can control it very well by simple filtration and certainly by water treatment. So the other thing that's interesting is the blood type makes you susceptible. I think it's the O blood type that, I think I've got that right. The O blood type makes you susceptible, which is why there's very little O blood type in Bangladesh. I hope I've got my facts correct on that. In any case, the blood type matters, and the blood type that is selected against because you're susceptible is much reduced in Bangladesh. So there is this, that's why I'm said earlier that we're taking into account the kinds of models, incorporating the models that have been developed for transmission and immunity populations that have been developed just by epidemiologists. And what we're trying to do, and I think, Mike, we were talking about this earlier, is taking a series of models and combining them together. Yes?
So now that we have this model, can you use it to predict future cholera outbreaks? Like, can you use it to predict the outbreak in Haiti? Have you been looking at rainfall for
The question is, would we have been able to predict the outbreak in Haiti? And my answer is yes, had we applied it. Because I don't have time to give you all the stuff we're doing. We've been doing a lot of work in Africa, in Senegal, and we've published some papers where the same phenomenon occurs. You get elevated temperatures, very heavy rainfall, and bingo, the cholera erupts. And so we can predict in Senegal and a couple of the other countries on the west coast of Africa, where they do have a lot of cholera. We've been able to apply the predictive model, and with historical data, hindcast, we can now use that for forecast of conditions that will be conducive to outbreaks in Africa. And the intent is that we develop the satellite sensing tool as a public health tool. and we could use it for malaria, waterborne diseases, and vector-borne diseases.
Is the World Health Organization or UN or anyone actively applying these tools to reposition antibiotics, that kind of thing, or is it still
Yes.
basic?
The question is, does the World Health Organization and other global health units looking into this as a potential mechanism for providing health care where needed? And that's precisely what we're hoping will be done, and that is to be able to predict the epicenter, if you will, of the disease potential outbreak, and then to be able to ring it with shipping in the antibiotics and the fluid rehydration, because, you know, it takes liters and liters of intravenous fluid, but fortunately an oral rehydration method has been developed. Interestingly, it was developed in India, but you now can buy that oral rehydration if you ever had a baby, a child with severe diarrhea, and you were told by the pediatrician to go get some Pedialyte. That is oral rehydration therapy developed to treat cholera and to replace the fluid lost. So, yes.
Are there animals that get cholera? And have you ever come across a person who's immune to cholera? I mean, they can drink as much fat water as they want, and does that have any effect on them?
The question is, do animals get cholera, and are there people who are naturally immune? In the first instance, the reason that we discovered the dormant stage is because, in fact, there's a book published by Pulitzer in 1947, WHO volume, where it describes hundreds of tests of cows, donkeys, chickens, all the domestic animals, without any success in isolating the bacterium. So it's an environmental bacterium, an aquatic bacterium, and not a domestic animal disease. The notion of treatment, it's really just providing safe water. If you have water that is properly treated, boiling water is quite sufficient. But the trouble is, in Bangladesh, there's not enough fuel wood. The women actually collect cow dung and dry it, and that's used for cooking meals and so forth. So it's in the case of the sari cloth filtration that we developed, which so cheap anybody can use it, it's an extremis solution. It's when you've got severe floods and you're up to your ankles in water, to be able to do something to provide safe water, or at least relatively safe water for your children. Yes?
Cole, just to clarify, Mike asked the question, are the World Health Organization or other global health organizations using the model? And your answer said, we would hope.
Yeah, the answer is...
So I infer, therefore, that the answer to the question is no.
The answer is no on an official basis, but some studies are beginning to be done. And the value of using satellite imagery for monitoring, particularly for insect-borne diseases, using the Landsat type data, have been used. But
This is fairly recent research too. You wouldn't
Yeah.
expect it to be adopted
Right.
This quickly.
Right, our papers have first appeared. The first paper on the cholera study was something like 1985, 88, so it's relatively new.
Do you have any estimate on the potential savings in lives if the models were employed globally with satellite sensing?
Well, the question is could we predict how many lives could be saved if the satellite sensing, remote sensing was used? I think in Africa it would make a big difference, a very big difference, because they're having some massive outbreaks in the Congo, particularly in the Sub-Sahel region. In the Mozambique has big outbreaks every year, and I think the sanitary conditions being what they are, being able to predict from the weather and expected extremes when the epidemics would be most severe, I think could be extremely helpful.
In the last year or so, they've been talking about putting a plastic bottle with water in it in the sun. Is that sufficient to deal with the problem?
There are a lot of techniques that can be used, and one is, as just asked, the putting plastic bottles of water in the sunlight. Actually, it's trays of water because UV doesn't penetrate thick layers, but it penetrates thin layers. So if you have just a small layer in the bottle and it's on its side in the sunlight, both the temperature elevation from the sunlight coupled with whatever UV would act as a disinfectant. And there are a variety of techniques. I'm working now with a group that I really enjoy working with. It's called the Safe Water Network. It was founded originally by the late Paul Newman and Joanne Woodward and several wonderful philanthropists. It's a tiny little organization, but they use a business model for building kiosks that provide filtered water, and at the same time they train people to run it, to operate it, and these are highly educated people with a good business plan, and then with a costing that is absolutely minimum, you know, like 7 or 8 cents per 20 liters, but it's enough to recover costs so it can be self-sustainable. And they have about 40 or 50 of these in Andhra Pradesh and another in Ghana. And I like the approach because it's a sustainable approach. It's a business model. It's not just philanthropy where you're constantly putting money in, but you're getting the people to own the operation, and the community develops pride in what they're doing. And so I think that's great.
So the one thing you did not do in your presentation, you are doing it somewhat in the question and answers, is the approaches or policy recommendations
Yes.
that you would make. So I want to offer you the opportunity to summarize all of them because
well, I'm being recorded, but hey, I've reached an age and stage I can say what I think. I wish we would invest in safe drinking water and sanitation, and I think the Gates Foundation is beginning to address the sanitation problem. Beginning. I've just come back from Stockholm Water Week, and the whole week was focused on how to bring sanitation, and it sounds crude, but how to bring facilities for sanitation as opposed to field use as a latrine, which is a big, big problem in the countries like Bangladesh and India and some other lesser developed countries. So if we could invest, and I don't have time to go into it, but we're doing studies using a new algorithm to be able to analyze what's in the stool of a patient coming in with cholera, and we're finding anywhere from 4 to 10 pathogens. In other words, they don't just have Vibrio cholera. 60% have rotavirus. Half of them have Giardia as well. They have a little dose of Salmonella and a little bit of Shigella, and you know, it's a mixture. And I think we're just beginning to understand infectious disease. And what lands you in the hospital may not be just that single agent that the symptoms seem to be correlated with. It may be that you get more than that, which is why when you get treated against the pathogen that is sensitive to the antibiotic, you treat the patient and you get a secondary infection. That's probably because that other pathogen's there and it's resistant to that antibiotic. So we need to move into what's now called metagenomics and apply that to infectious disease as well as well-being, which is being done. It's a very exciting time, really. I think I've worn you up. Well, one more.
Just a question about microbiome, and as we're learning more about, positive bacteria and
Yes.
probiotics, has there been any work in terms of understanding, as we're starting to understand a little more about, a gut microbiome and how that might be leveraged to treat or, help individuals?
The question is, how can you use microbiome knowledge or information to treat enteric disease, or for that matter, any disease, maintain well-being? Well, it turns out that currently, because of overuse of antibiotics and the fact also that it's just a really difficult bacterium to treat, Clostridium difficile, it unfortunately is very common in nursing homes, and also it is pretty lethal for babies as well. And it's very hard to treat, but now one of the treatments is to introduce normal bacterial flora into individuals, either through probiotics like yogurts and so forth, which has been used a long time. But now, sounds a bit gross, but using fecal transplants from healthy individuals, and it's been effective against Clostridium difficile, which is a killer. It's a debilitator, that's for sure, and a killer. So the more we learn about this mix of microorganisms that's in our gut, on our skin, in our oral pharyngeal region, to the point where some scientists are referring to the microbiome as another organ of the human body, like the liver, the heart, the lung, etc. So stay tuned, folks, because it's a wonderful world we're living in. Things are happening, particularly in the area of microbiology and the microbiome. Thank you very much. I've enjoyed being here. You've been terrific.
Once again, ladies and gentlemen, thank you for coming. I hope you'll come to our future CReST Bold Idea events here at the Potomac Institute. The next one is on the 20th of September, where we'll look at global climate change impacts in the Pacific. And you're all welcome to stay. We're going to have a little bit of wine and some snacks, and hopefully continue the conversation in this room. So thank you for coming.